Our next speaker is Dr. Karen Mohlke. I met Karen when she took Mike's 666 class. She was a graduate student in human genetics, and I thought, wow, this is interesting. What's she going to do with this? And then she went on to do a postdoc with Francis Collins, and I thought, wow, that's interesting. Look what she's doing with this. You know, she was one of those people that said, we can do this better every time. Like here's some information we have. We can do it better. We can understand it better. And so she has been a real force in fusion in that way. And I can't wait to have her talk about it. She's been in there from the beginning. Karen is a professor at UNC and vice chair of research in the Department of Genetics. So we're really happy to have Karen here. Thank you, Karen.
All right. All right, so thank you, Beth, for giving that introduction to explain what I'm doing here. So I was a human genetics graduate student with Dave Ginsburg, and I was crazy to take that 666 class, but Mike is so kind and generous that it was okay, and I managed to fumble my way through and learned some statistical genetics along the way. And learned how cool it was, even back in this era when positional cloning was just happening, that it was possible to move into complex traits. So I was asked to give the fusion retrospective. So fusion is now 33 years old, and so I'm going to talk us through a little bit. You've heard smatterings of this through different presentations the past couple days. So some of this you'll have seen before, but I'm going to try to tie it all together. And this top picture there is from a month ago, the most recent time that the fusion collaborators got together. So I'm going to start with the origin story. And I was not around for the origin, but I have heard the origin story told many times. So this is going to be like the telephone game of me trying to remember and put some pieces together and all. So my understanding is that Mike and Francis had been working together, and they had been doing mapping. Mike was doing a lot of radiation hybrid mapping. They had been mapping single disease genes, NF1, had worked on BRCA1. And they got together and they said, what's next? What should we next be working on? What's the next challenge? And they decided that for a complex trait, and they came up, of course, right? They came up with the criteria by which they would make the decision about which complex trait that would be. So they thought it has to be genetic, has to have some heritability to it, common enough that you can go collect patients, participants. I don't know what all the other characteristics were, but they came up with the list first, and then they both went off to think about what potential diseases could that be? What should we go study? And as I understand, they both came back and said, non-insulin dependent diabetes mellitus. They both had independently come to that recognition. So great. Okay, so they have an idea. They've got a trait. Where are they going to get the samples? So the next part of the story, as I understand, is that Mike was in Finland. And he was giving a talk. Probably this was a radiation hybrid mapping talk. I don't know. He was interested in genetics. And he met Jaakko Tuomoletto. And Jaakko had been doing clinical research, the epidemiologist, doing clinical research in Finland, had set up 50 sites, working with participants. And Mike gave this talk, and they met at the meeting, and then they want to talk a little more, and Jaakko says, why don't you come over to my house? We can have a sauna together. So Finland, it's a country of 6 million people and 3 million saunas. And so they met together in Jaakko's sauna, and I'm pretty sure ChatGPT thinks that they met in an American sauna. I think in Finland might be a little bit different from this. But whatever, they had a good conversation. I think they talked about DNA and diabetes and came together with the idea of how to go forth and work in Finland and collect samples and to be able to study diabetes. And then they said, okay, now we should get some physiology in this mix. We need to understand the basis of disease. I had recognized they wanted not just to be studying type 2 diabetes, yes, no, but those related quantitative traits. Now my understanding is that Mike had been teaching Rich Bergman's minimal model. And so together with Sumitra Ghosh, contacted Rich Bergman, who every time we meet says, I'm not a geneticist, I play one on TV. But he jumped at the chance and got together and met. And so that was formed the beginnings of the fusion study, creating this collaboration that has now been ongoing for more than 30 years. So fusion set out with this relatively simple question: what's the genetic basis of type 2 diabetes? Really, where is the diabetes gene? And 33 years, and I gave up counting, so I just rounded and said 330 plus papers, a field transformed. Because so much of the process has been the statistical methods development that goes along with the data generation and all. So I want to bring you back to the early 1990s and where we were. Type 2 diabetes, non-insulin dependent diabetes mellitus at the time was known to be heritable. We heard what the lambda SBS was from Steve Rich. The primary defect was uncertain. Was it insulin secretion? Is it insulin resistance? What's the relationship? How is this? Which one comes first? How does it all happen? Don't know. Most of the genetic studies that had been taking place were the candidate gene style that we heard about, and we heard how poorly those replicated over time. And linkage analysis with an affected sibling pair design, helpful because, right, if an older age onset disorder, you don't necessarily have access to the parents. But an affected sibling pair design had worked for Mendelian disease to identify genes. Great. So there were some challenges at the time, and that continued for some time. So diabetes, a very polygenic, heterogeneous disease. Didn't know quite the degree of that at the point. Environmental influences affect risk. Families were small. The human genome was not sequenced. There was no physical map, and the genetic map wasn't really done yet. It was like on the way. Genotyping was low throughput, and there were limited statistical tools to deal with all of the challenges that came along the way. So, but those genetic maps were getting better and made it seem possible to be able to do genome-wide mapping. So we could say no to candidate genes and say gonna go genome-wide. At the time, with positional cloning, there was a recognition of how the allele frequencies were different in different subpopulations, and so there was potentially some benefit to the Finnish bottleneck. Turns out that benefit comes, decade or so, a couple decades maybe later. And so Mike led the design here of how do we maximize the power to detect linkage for type 2 diabetes? And this was a very well thought out, think through all the different possibilities, set this up. So the probands had to have a pretty early diagnosis, increase the chance of genetic risk in the individuals. I figure half the people haven't learned about linkage analysis because it was like passé by the time you're here. So, okay, proband early diagnosis, increase the chance that it's genetics. Use this affected sibling pair design so that we didn't need to have large multi-generational families, but still try to get large families, more individuals. They aim to minimize the chance that the risk was coming in through both parents, which if you had like a major gene, maybe that would be true. Finland had a lot of type 1 diabetes, so there was then extensive, how are we going to phenotype, how are we going to carefully figure out that there are not type 1 diabetics making noise in the data for type 2 diabetes, and then to do some extensive phenotyping. And in the study design paper that was then published here in 1998, we forecast how this was going to go. 400 affected SIP pairs typed on a 10 centimorgan map, there should be 82% power to identify a gene that confers 1.8-fold risk with the log square of 3.
So come on, only 50 SID pairs were needed for type 1 diabetes discovery. It should have worked.
It should have worked. It should have worked.
HLA and type 1.
Yes. So thus was launched the fusion study, Finland, United States Investigation of NIDDM genetics. I guess sweet genes was in the second running for the abbreviation, and fusion won out. And then when NIDDM became type 2 diabetes, it seemed like the acronym would get a lot more awkward with a T there instead of an N, so it just stays. So this was a major collaboration then set up with these 4 main sites, focusing on doing the subject recruitment and clinical testing was happening, based in Helsinki, but with recruitment happening around the country, DNA going to Francis Collins Lab at the NIH, and the plasma samples going to USC to be able to measure like glucose levels and various quantitative traits. And then Mike basically tying the whole thing together and leading the statistical analysis of all this work. I'll say now, Mike just calls himself the coordinator of fusion, but he's the leader of fusion, so very much. Okay, so here's some of the first and people in the room who participated from the Michigan side in some of these initial analyses going on. And Mike had many roles, has many roles throughout the fusion study. So, of course, he's the statistical leadership behind this. And I'm going to talk a little bit more about this later, but so many methods got created, developed, things figured out because of the problem of what's happening with the fusion study and what do we need next. Aspects of the study design, starting with what I showed you with the affected SIB pairs, but then I'll show you the transitions that happened over the 30 years. And there's a study design question constantly popping up every 5, 10 years or so in what do we do next. Mike has this impressive ability, as you've already heard, to be able to tie people together with different kinds of expertise, get everybody communicating, not talking in words that everybody else isn't understanding. So that level of being able to integrate the scientific disciplines, integrate the trainees from across the different sites into being able to participate actively, play roles throughout the process, and leading the team interactions. And so in the beginning, well, I don't know the beginning, but in the middle, teleconferences, lots of regular teleconferences happening, and then the one-on-one, have to have face-to-face meetings. This was a key, critical part throughout the project. Those face-to-face meetings involved a bunch of science, but also having fun and being able to interact with each other, get to know each other a little better. It makes it a little easier when you go play games together to then be able to ask somebody what you think is a dumb question about this aspect of the project across the way. Lots of music happening in those gatherings. When Fusioneers were asked last month, Fusioneers from across the years, for their reminiscences, what did they remember the most? The gatherings, especially the gatherings hosted by Mike, was like 80% of the answers and stuff. So like the interactions between the people has been key. Okay, so next I'm going to go through an evolution of the fusion study and talk through some of the progress that took place. So as I've already said, this started with an affected SIB pair linkage analysis with some microsatellite markers. Whole lot of mom, dad, the kid, make sure that the kid's inheriting markers from mom and dad. Whole lot of work went into hundreds and hundreds of microsatellite markers, many individuals, and checking for the genotype quality. Then the beginnings of this collaboration, the collaborations that have come to, what type 2 diabetes is today. I went and pulled and looked at what the dates are. Look at the dates for when the linkage consortium that Nancy and Mike got going. That consortium agreement letter of participation, 1997, and who are the participants then? Sort of building up already very early this idea of we have data, we're going to have data, how are we going to be able to work together? And I think this really sets the standard. If you think about where different other physiology fields, other disease fields, and where they were in terms of working together, type 2 diabetes was at or near the start of that, thanks to this. There was a brief foray into candidate genes, because I'm going to come back to some of the discoveries, but there was a brief foray into candidate genes, and I'm reminded of this stage when it was too expensive to genotype people on individual markers. So we pooled together the cases, pooled together the controls, quantified the alleles to look for differences and potential evidence of association. Thank goodness of technology has progressed since then. Then we have linkage, and we have some candidate genes, and then really we've heard about this already, but Rishmir Kangas, and this idea of maybe association studies having more power than the linkage studies do. So then that led to a genome-wide association study. So the remarkable thing I think that happened at this point is, how did we get genome-wide genotyping done? Mike, and maybe there's some Francis in here, I don't know, but somehow fusion became the test case for CITER, the Center for Inherited Disease Research, to be the test case for which they could run the very first of the Illumina genotyping arrays and see whether they would generate good quality data that others then might want to go do something like this for their samples. So 2006, probably a little early, 2005, 2006, is the time when then CITER took on doing the very first of the genome-wide genotyping with Illumina arrays to enable the GWAS to happen. And it didn't take all that long to realize that the sample sizes that existed weren't going to be large enough. So there was already a second stage of linkage analysis that was happening, Fusion 1, then Fusion 2, and then it was called Fusion and Friends, and it was finding any other samples, other people, other bring some more folks in that had other cohorts, right, that had been collected, were available, for which genotyping could be happening, so that we could expand the sample size and learn more about potential risk alleles for type 2 diabetes. I want to point out this one, is this going to work? This one group here. It's actually, I think it's shortened here. It says elder controls. Really, they were called the elderly controls. This was to have a normal, to be able to look at what the allele frequency was amongst individuals that were not, presumably not at risk for diabetes. So how did this get defined? I looked it up. It was individuals who were born in 1925 and who in 1995, you do the math, 1995 still were normal glucose tolerant. Okay, so then those got called the elderly controls.
We don't call them that anymore.
It only took like 15 years or so, and there was a moment in a fusion meeting that it was collectively decided that they were not the elderly controls anymore. They were just older controls. Okay, and so then more and more, get people together. How do you get more people together? So you heard some of this already from Mark, but this Mike and Mark and David Altshuler getting together and saying, hey, we should be thinking about combining our data, our genome-wide association study data together, and then building from there. So Mike playing a key role in like the glue, getting people to work together that would maybe otherwise see themselves as competitors, but recognizing the value. So this happened for type 2 diabetes, but also Mike helped get people together to study anthropometric traits. So that's that forms the GIANT Consortium, and the glucose and insulin traits, and that forms the MAGIC Consortium. And I don't think it got called global lipids genetics for like another cycle or so, but basically getting together across the different traits to be able to do these genome-wide association studies for many related traits. Now that we knew that it was possible, what to be able to do next. And then came exome and genome sequencing. So I'll start with the metabochip stage. This was when it was still too expensive to sequence, but some sequencing had happened, and so there were some low-frequency variants known, some regions that we wanted to sort of do a better job at, collect that together. So sort of across the different consortia, put together a plan for genotyping that Illumina would create, and then it would be cheap for everybody to go apply this to their different studies. And then sequencing. So this is the Go T2D study and the T2D genes study, gathering groups together. Nancy and Mark is included here, and figuring out together how to design the next stage of exome and genome sequencing studies. And they did lots of work thinking through all the different ideas and possible plans. And by the time they were done, it was cheaper to then shift and do something even more deep and more extensive to be able to generate sequence data. Fast-moving field. Okay, and then there's a stage of saying, we're going to need to learn more about these loci and how they work and what they do, and we really need something other than blood. How are we going to study this something other than blood? And the discussion started, and they asked Jaakko, say, if we asked some of the participants to come in and give a tissue sample that's from their fat and from their muscle and get some fibroblasts, we'll be able to make some iPS cells and be able to differentiate them. What do you think? Are people going to be willing to participate in this? And he said, yeah, like 70% of people will say yes to that. We're like, oh, this is why we're doing this study in Finland. So not just can we get some people who are willing to participate, but how about can we get some people who have normal glucose tolerance, some with impaired fasting glucose, some with impaired glucose tolerance, and some newly diagnosed type 2 diabetics, because you wouldn't want to, if you knew somebody had diabetes, you wouldn't really want to be doing this level of intervention. So getting individuals in these different categories so that we could have tissue from a range of phenotypes relevant to type 2 diabetes. And then that has then moved on to, and sort of where we are today, the current status is integrating cell type multi-omics and learning about the mechanisms. Okay, so as I said, a lot of this involved lots of get-togethers, and it will say, to my memory, it's like 90% of them were at Mike's house. This is Mike's backyard. I was like looking through the pictures to see how the backyard changed over the years, this is in 1999, which is sort of the before that even that first linkage results were published. Okay, so I have now 3 themes of progress that happened sort of across all of these years. I've sort of categorized this into 3 themes. I mean, if we really wanted to talk about what fusion has done in 33 years, we would be here for a week or 2 or so. So this is just sort of selected examples across the way. So the first theme is mapping genetic risk. And I wanted to go back to, this is sort of, I would say, the first main result of the fusion study, the first time we were like putting out what happens in type 2 diabetes. I left the axes off on purpose so you could think about what you might imagine this is. I first looked at this, just recently and thought it definitely looks like chromatin accessibility to me, like a 1 kb, 1.5 kb region of the genome, maybe. And I'll give you the hint that this was published in 1999. And it was the first affected SIB-pair linkage analysis, the major highlight of what we had done. And you'll see that most of chromosome 20 is covered by, because what we we gave up on the LOD score of 3. And so it was sort of take the peaks and go down a megabase, go down a LOD score unit and consider any gene in these regions that within there as a potential candidate gene for type 2 diabetes. So the early signals were imprecise. And we had meetings where we went through every phenotype that was analyzed in every linkage peak and many different types of analyses to maximize. I mean, there was a lot of work that had gone into this, so let's maximize the chance of identifying things. And this is looking for genes under the linkage peaks. I remember especially one meeting, it was held in Airlie, and we had divided up the chromosomes into groups, and we'd made groups of people that would look through the genes and talk about what the potential mechanisms would be. And I would say the main outcome of that meeting, in my eyes, was you can make up a story about any gene you want, and it can play a role in diabetes. Okay, so thank goodness. Then 14 years after starting the fusion study, genome-wide association study, and I pulled out this variant here, TCF7L2, stage 1, p-value 10 to the minus 5th, stage 2, 10 to the minus 4th, combine them together, 1.3 times 10 to the minus 8th. This, and you've already heard from Mark, right? This study, and then the study that Mark led, and the study that David Altshuler led, and the 3 studies together combined compared results. One of the things I also want to point out is in this first, so this is a region, this one's in chromosome 3, this IGVF2, BP2, locus still very strong, that already at the time when there were 10 signals identified, already we were talking about, well, if this is your association signal and you take a variant to represent it, if you condition on that variant, is there another signal nearby that's playing a role? And at the time, no. This was the entire signal is this one, do this one thing. But that's a theme that then persists and grows over time because the sample sizes do get bigger and there do start to becoming multiple variants, multiple signals near each other. Okay, so then what did this paper have to say? So first of all, there was the comment, these are quotes from the paper, that now that there are 10 robust and replicated association, this is a landmark in the efforts, and it truly was. And these 3 studies together, and there were a couple others, all 2007 or so, really represented this landmark in efforts to identify variants that are associated with any complex disease, but here type 2 diabetes, and also that it's likely that there are other additional T2D predisposing loci to be found. Prescient. Also, in the discussion and towards the end, and I just need to start by saying Jim Neal was the founder of the Department of Human Genetics here at Michigan. And his opinions were well known. So think about the bravery of Mike and Francis at Michigan choosing to study type 2 diabetes. Because 30 years before, Jim Neal had labeled T2D as the geneticist's nightmare because of how challenging it was going to be, because of the genetics and the environment and how this was going to go forth. And they noted that until recently, his prediction has proven true, but that even though a lot remains to be done, we are at last awakening from Jim Neal's nightmare. So this was a wonderful revelation, this project. Many millions of dollars had gone into it to this point, and now are able to start talking about associated loci. So really, this point in the story is a paradigm shift. Right? This discovery, complex traits are highly polygenic. Of course, we know this now, but at the time, that was a big deal. It wasn't that we were finding one locus, it's that there are many. Instead of a few major genes, many small effects. And even starting to look at the biology of these genes, we were learning new things about, so the gene nearby, and go look at its biology. And so, oh, there's a zinc transport, intracellular zinc transporter, and how is that playing a role? And learning about the pathways that were contributing. Finding that a lot of the first genes were involved in beta cell insulin secretion, right? Starting to be able to look at that, insulin secretion, insulin resistance. What are the contributions? And even that first paper did some polygenic risk prediction. Take those variants together and look at what the difference is in terms of disease outcomes, the disease risk based on those 10 genotypes. So then, very soon after, for type 2, we moved from type 2 diabetes to these consortia that are looking at other traits. So this is already by 2009. Here's the example of, again, groups coming together, anthropometric traits. This is body mass index, and beginning to identify novel loci for that trait. Lipids by 2010, 46 or so groups had gotten together. So more than 100,000 individuals in those different cohorts combining data together. This is a pretty fast shift from 2007 to 2010, getting this many groups to come together to start identifying and contributing to the number of loci identified for the different traits. I'll go back to type 2 diabetes. Right, time progresses. There's different rounds of GWAS meta-analysis. So continuing my theme of the discovery, right, fusion is contributing to these different studies as the years go by, and this is the number of loci identified and the contributions in the different populations that are playing a role. And getting up here in 2022 at 534 loci. And then this is the most recent published type 2 diabetes GWAS meta-analysis. So we've grown to now 2.5 million individuals, 1,200 or more association signals at more than 600 loci. So plenty of examples of 2, 3, and more association signals in the same region. Here, chromosome 10, so here's TCF7L2. Now we just have to crop the y-axis for those p-values and stuff. Joy to see. Okay, so theme 1, mapping genetic risk. Fusion helped move the field from where is the diabetes gene to how do hundreds of variants shape risk. Theme 2 is now the statistical methods needed along the way. So there was a lot of them. I had to use ChatGPT to help me out. I took lists of Mike's papers that I knew were relevant to the methods developed for fusion along the way, and I asked for groupings and things along, and also for some pictures. So I'm going to attempt to do an example of each of these, but you all know better how these methods came about. So first category here is pedigree analysis and linkage mapping. So there were many years of linkage analysis methods, and I just pulled out snippets mostly from titles of papers that are showing the kinds of questions that were being asked that were needed to be addressed to help understand what to do next, how to interpret the data, how to do the analysis. Lots of issues you've heard about, Mike's accuracy and importance. So thinking about what happens if you have map uncertainty, you get it. Beth, I very much enjoyed the ordered subset analysis, different ways of making good use of that linkage data to try and understand maybe there's groups of people that are more similar in phenotype that have genetic risk contributing. Here's a result from Mike Epstein that I think he published a while after he graduated, but with Mike and with Zhihong, looking at variance components methods and addressing like not always do the families fit the assumption of normality, and so what happens if they don't, and can we have a method that will allow for a broader application of methods to be able to make better use of the data that's available. So across them, I would say the methods refined the mathematical rigor of the linkage studies in this time. Another category of methods development is genotyping and study quality control. And we now know from Mike's earliest paper with HLA that he was very aware of genotyping error, and so that this played a big role, right? And now we all assume that the genotype, there's some sort of very standard checks you do, and to make sure that the genotype state is high quality, that the samples are good, that the SNPs are good, that all these various steps. But it came with trial and error and methods to detect when things weren't going quite the way they should. So a series of methods here, and I'll pull out one from Julie Douglas, and it's looking at the probability of detecting error given no genotyped parents. And this is back with the microsatellites. So even if you had lots of multi-allelic markers, but you didn't know the parents, what's the chance you could figure out if there was an error in that microsatellite data from a given family? And so what this led to is being able to figure out which families do we either need to resample, or which families do we remove from future analysis because those relationships are potentially not what you expect them to be. Clean up the data. So I'd say across this sort of set of methods, these and tools, they are tools that helped ensure that the findings were due to biology and not due to experimental error. Third category is thinking about GWAS design and meta-analysis and interpretation. And so multiple, right, lots of questions being asked. How should we're changing now from doing linkage to doing association analysis, have these different samples, cost still matters, who can we afford to genotype on what, and then what do we do next, and how do we make the decision about what to do? Gonzalo already talked about this paper, so I'm going to use it as so this is the, when you have the Fusion 1 samples and you have genome-wide genotypes on them, and you're deciding what to follow up, how many things do you follow up, in what size sample, how much does that matter? And the answer, thank you to Andrew, Skoll, and Gonzalo and Mike, is you should be doing, don't do this replication business, do a joint analysis. So the point I want to make is, right, this is the data communication point. So this paper has to be written not just for the statistical geneticists down the line that are analyzing the data, it has to be written to the people that are deciding what to genotype and how big their study needs to be. So I love these black bars here. This is the what you're missing piece, what you're missing when you don't do that. How clear can you get to help even somebody like me get the point very quickly of what the best approach is? So I very much appreciate the data communication lessons I've learned from Mike. So, but this fundamentally changed how large-scale genetic studies are designed, that there's not so much do the study and then go do a replication, or how do you combine that? Do the bigger study, combine the data, which is now the way that the vast majority of association studies then are done. Okay, and last category is the sequencing and rare variant discovery and design. And so this is moving into the exome and genome sequencing. We have our fins, and thinking about sequencing imputation and cost-effective strategies. So my example here is here from Adam's paper, and it's looking at sequencing and alleles and comparing and showing the benefit of using a population isolate. And so the green dots are representing what the allele frequency is in non-Finnish Europeans of these different variants, and the blue dots are representing the allele frequency observed in the in Finns. And the paper is providing a power analysis for future use and understanding of the role of using a population isolate and what the power might be. And then I like this figure for the This is showing the location of the individuals, not their birthplace, because people move around. How about their parents' birthplace? Their parents back before there was so much moving around Finland. So now you get a better view of what the migration patterns were longer ago, back representing perhaps how a variant arises and then moves through a population. So here the red is representing the individuals that are carriers for one of those, I guess it's orange, carriers of one of those variants, and the blue is representing an equal number of parents randomly from the non-carriers of this variant. And so you can clearly see that how the past has led to the discovery of this association. So statistical tests identify rare variants that were often missed by standard GWAS, so expanding the discovery of associations. So through these statistical methods, Fusion didn't just produce results, it helped define standards for credible results. And I'll say one of my greatest joys in the Fusion study and going to these annual meetings was watching, like we're trying to solve a problem, like discussing something, a question comes up, usually, Francis asks a question, Mike looks, he thinks about it, stuff, and he's like, huh. And then, okay, me, fast forward a year, it's a student's thesis project, and the student is presenting and showing us the answer and helping us know, okay, this is how we should be addressing this question, and this is the strategy. So I know a lot of work, all of you, a lot of work went into those analyses, but it was very fun to see the back and forth between the question and the how do you go do the statistical analysis to figure out what the right way is to go forward, and that it just continued throughout the project. Okay, and my last theme is going from association to biology. So the different strategies of getting from that GWAS signal in the first place to understanding how it could play a role in type 2 diabetes. So a few different things here. So first, I'd like to better highlight the metabochip. Again, it shows the bringing people together for the common good. So here are the different consortia that Mike and others wrangled to contribute to this, creating this array that would make it more cost-effective to follow up on additional variants. So strategy was people might want to be fine mapping, figuring out what are the variants that were not tested, some of which might be lower frequency, the variants that were not tested in the regions around the most interesting associations. But then also, those associations that were not quite strong enough, could we boost the power and more samples? So everybody pick 5,000 or so variants that you would like to follow up, pick some regions, I think they were gold, silver, and bronze, for the density of the variants that are going to be contributed, and to demonstrate the value of imputation accuracy, figuring out what those other variants are given the density of variants possible on this next level array. Also some fine mapping that used imputation and epigenome maps. This is now Anuba Mahajan together with Mark and others, combining the data together and evaluating the signals that can be identified as you move into lower frequency variants. And so you can see in purple now the novel T2D loci, and the asterisks compared to the dots are representing finding more than one association signal in the same, at the same locus, in the same region, so another genetic impact on some particular gene. So it's showing this discovery of these second signals, moving into the lower frequency risk alleles, improving the fine mapping, getting us closer to what the causal variants might be to figure out what the genes might be. And there are coding variants under some GWAS loci, so a focus on coding variants to understand what the genes might be. So here, this is showing work from Christian Fuchsberger and many others in the room, looking at coding variants possible through looking at the sequencing data and identifying these various genes, some of which are known to be driving a common variant signal, sometimes some outside those known common variant regions, some in tight LD with a non-coding variant, being able to sort of characterize these, identify some variants that might themselves be directly responsible for those evidence of association. And this is nice, but not a very large proportion of the overall signals that have coding variants that may be responsible for them. So then that fusion tissue study comes into play. So now the collection of the muscle tissue, the adipose tissue, the fibroblasts that can become iPS cells and differentiated into beta cells from different individuals. So it was then, and still is, pretty rare to have individuals that have both extensive phenotyping and gene expression measured, and genetic variants across the genome. But that allows questions to be asked more thoroughly about comparing things like type 2 diabetes status to expression level of genes in, this is bulk muscle tissue, and being able to start getting at some of these pathways that are, say, in individuals with type 2 diabetes have lower expression of various types of genes that work together in pathways, start to understand how some of those variants may be acting through muscle tissue to impact diabetes risk. And we also have genetic variants and gene expression across the individuals. So now this is an example of identifying type 2 diabetes variants at a region where in muscle the variants are also associated with gene expression level of a particular gene. So here the lead variant is a type 2 diabetes variant, and what's shown is the variants in this region that are associated with expression level of ANK1 in muscle tissue. So this is one of a small number of the strongest variants in this region where the ANK1 gene is located, and we can now take this data together with evidence of regulatory marks across in different tissues and comparison across different cell types to recognize things like, so amongst this set of variants, you can see that there's this, tends to be orange and red here, red representing a potential promoter, especially in muscle and a muscle cell line, compared to some other cell types. And at higher resolution, go look at that particular variant, indeed in a likely promoter region for muscle cell types, and then be able to make a prediction of that perhaps the transcription factor is binding differently to those two alleles. So here's a band, one allele and not the other allele, and it's likely this transcription factor or a relative because you can sort of shift that band on a gel to demonstrate it. So now we're to the level of a genetic variant, a transcription factor, a potential gene that could be in a tissue that could be influencing risk. The other cool thing that can happen with those tissue samples is not just looking at gene expression, but how do variants have an impact on gene expression and in what cell types? So now this is data, Arushi Varshni, Steve Parker, using those fusion tissue samples and looking at individual cell types. So take all the muscle cells, cluster them by the cell type based on their gene expression pattern, and now let's go look at the chromatin accessibility, whether it's opened up to let transcription factors in to bind at a given position. So there's three examples shown here. One is this region, so we're zoomed in quite tight here. This is like, hundreds of base pairs, and it's colored by genotype at the position of the dashed line. And so you can see that in type 1 fibers here that there's a different, the people who have TT genotype don't have very much chromatin accessibility, where the people with CC do. So CC more open, more able to let a transcription factor in and act. And this particular example, that region that is identified pretty similarly across all of these different cell types within the muscle tissue. The middle one's a little more selective. It's in some cell types, but it's not detected in some other cell types. And the third one shows a lot of specificity for a given cell type, these fibroadipogenic progenitors here. And so there's a variant in this region that is only detected in this one cell type. So now we can go from the variant to the regulatory element to potentially to a transcription factor in a specific cell type that can be influencing disease risk. I don't want to leave adipose behind. So adipose, the fusion study has bulk, adipose, subcutaneous adipose, EQTL data. And I want to talk about quality for a moment here, because if you just thought that identifying variants associated with a trait, in this case gene expression, it was dependent only on sample size, note here that the sample size for fusion is the lowest compared here. Fusion is green. Over here, which study identified the most EQTL genes of these 5? It's fusion. Which one has the most signals? It's fusion. It's not only the sample size, it's the sequencing depth. It's mostly not that either. It's the quality of the data that ends up having a quite big effect on discovery within the EQTL data. So this is another example of you don't just find one association signal, especially variants that can affect a gene. You can find 2 and 3, right? There could be variants in the promoter, another enhancer, a silencer over here, contributing to an effect. Thanks very much to Mike and Ji Hung and this wonderful APEX package that allowed us to do meta-analysis of EQTL signals across the multiple studies and be able to characterize them separately. So you can see then that in the meta-analysis, the sample size, the number of discoveries, both the number of genes that are identified and the number of signals are identified is greatly increasing. All your EQTL studies are too small. They're only identifying a very small portion of the EQTL signals that exist. This is the beginning, but there still needs to be more discovery to better characterize the variant effects on gene expression. Okay, this was bulk tissue. Oh, wait, no, let's make use of those EQTL by looking at whether the EQTL signals analyzed individually away from the neighbors that might also be having an influence on a given gene, and comparing them to the GWAS signals for various traits. So a given trait here, the number of times where a GWAS signal and an EQTL signal were identified to be statistically shared. So then you can start predicting that potentially the variants might act through that gene to have an influence on the trait. So across all traits, not all of which are shown here, this strategy predicts 1,800 potential candidate genes that could be playing a role in type 2 diabetes risk. So more than 1,800 genes, only a third of which are the closest gene to that GWAS signal. The negative being that you start discovering that a lot of those GWAS signals are co-localized with EQTLs for more than one gene. So EQTLs are not the only answer. There needs to be some functional follow-up to figure out which of those genes is playing a role. And including looking not only at the first EQTL signal, but the other EQTL signals, and looking at them separately and doing co-localization that assumes you're only looking at one signal at a time, increase those numbers of co-localizations with disease by almost 50%. So being able to go in and characterize the data most carefully allows more discovery. And then adipose data also is made up of multiple different cell types. And so the cell type-based EQTL data helps us solve and understand some of the challenges that have happened with bulk data, trying to compare bulk GWAS and EQTL signals. Let me try and explain with an example. So this panel up here shows GWAS data for blood pressure. Now there's 2 association signals in the region, so they're not colored by rainbows. One is colored by a red variant, and its LD buddies are different shades of red, and the other is colored in blue variants, and its LD buddies. Actually, there's only one GWAS signal here. And then in bulk adipose tissue, the EQTLs for this gene, CLIC4, there are 2 different association signals. So one GWAS signal, and it co-localizes. These are the same variants. Those red variants are the same as these red variants here. In CLIC4, it regulates endothelial barrier integrity. That's endothelial cells, the linings of the blood vessel. So People complain, though, if it's the weaker of the EQTL signals, the second one that co-localizes with the GWAS signal, why doesn't the stronger first EQTL also co-localize? If you're going to claim that the variants are acting through the gene to have an effect on the trait, then if you have two signals that affect that gene expression, shouldn't they both affect the trait? With at least 300 examples. The answer is cell type. So here is the endothelial cells. Remember, that's where the CLIC4 variant is acting on endothelial barrier integrity. The EQTL signal in the endothelial cells is the red signal. The blue signal is not significant. What about the other cell types present in the adipose tissue? Here, the adipocytes, very strong evidence of association, the blue signal, and the progenitor cells to the adipose, these precursor cells, also very strong evidence of association. So what we had been looking at in the bulk data is a mishmash across the cell types, and now when able to look at them individually, you can understand the basis for the association. So there's a presumably regulatory element acting in the endothelial cells. That's where the one or more of these variants is having its effect, potentially then leading to this influence on blood pressure, whereas the genetic effects on CLIC4 and these other cell types are irrelevant to the effect on blood pressure. So across this range of studies, fusion helped answer hard questions, moving from where an association signal is into what it does. There's one unifying aspect to those themes, to this whole process. One unifying aspect of the fusion study, you've heard it before, is the people. And it's the people that are participated, really the acknowledgements are probably everybody in this room participating in these projects. And I'll say that there's two aspects to the people when I think about Mike. There's Mike's pedigree and the people he's trained and who have gone on to train others and the relationships between those. But there's also the bringing people together aspect. They're getting groups of individuals working in different places, working on different studies, getting them to work together for the common good. So Fusioneers through the years, many different people participated in this project. So I have some photos here from 1997, very early on, the process of still setting up the study and starting to get that beginning data. You can see the four PIs at the time as they're working together to design the next stages. Here we are in 2002. 2002 means that there's linkage peaks. We're fine mapping linkage peaks with microsatellite markers while the human genome is being developed and we can figure out what markers are present in different places. So thank goodness that there's times to get together and enjoy each other and think about the potential and the science and what next steps to do. Moving on here into 2007, now there's something to celebrate that we have that first genome-wide association study result and are working towards what's next. What does this all mean? And then what's next? And I didn't add, there's like missing Fusioneer pictures in the middle here. So I just went and grabbed pictures of collaborators and students and participants in the project and that helped move forward all of these various aspects of the different study. And now here in 2023, very much moving into the how do these signals work? What do they do? How do we understand the basis of diabetes? So I'm going to end with a few reflections on the 33 years of the fusion study. And I'm going to start with, think about what changed in 33 years in technology. So as I understand, a couple years into the project, Jaakko said, we should check out this World Wide Web thing. It will be a lot better than faxes. The capabilities have increased, where we were in storing data, the amount of data how do you get around? How do you map things? Where are those people from? And how we interact, from the teleconferences where you didn't know what people looked like back then when you had teleconferences with them. You went to that one-on-one, the face-to-face meeting and went, oh. So what are 33 years of advances in diabetes genetics? So from a few known loci to more than 1,200 association signals, from linkage analysis into GWAS, large-scale sequencing. Now this is a collaborative global enterprise, the genetic basis of common disease. We know now not to be looking for the large effects, but to be considering polygenic risk. And really even starting to get at, is T2D a single disease or a heterogeneous combination of potential diseases, and being able to use genetics to start identifying the different clusters of how those variants might be acting in different ways to influence risk. 33 years of advances in resolution, going from a region of interest that basically covers the entire chromosome, 100,000, 100 megabases of region, to what these are single KB regions, and really focusing on something of a couple hundred bases wide as to region of interest. N scale is critical. You've heard that before. We've learned it through once fusion became fusion 1, fusion 2, and then fusion and friends, and then collaborations, and in through sequencing and into the role into genetic studies of other molecular traits. And then finally, great people are key. We've said that all throughout these 2 days that we've gathered together. It's long-term collaboration that made fusion possible. It's this focus on collaboration and not competition. It's learning that these face-to-face interactions are essential for the community building aspect of working together. And this is Mike's quote: "Life is too short to work with people," well, I'll paraphrase, "you don't really like." And I'll say that Mike's leadership has been essential. So on behalf of myself, on behalf of Fusioneers, many of them throughout the room, on behalf of human geneticists, whether they realize or not how much they're making use of the different statistical methods you've developed over all of the years, I'd like to say thank you. Thank you for your kindness, your leadership, your ability to lead with respect for all, and that really helped make this project a success. So happy birthday, Mike.